Students, in the first chapter of biotechnology, processes and principles, we have seen how recombinant DNAs can be synthesized. And here in this chapter, we will study about the application of biotechnology. By now, we know biotechnology has got immense application. Starting from the field of study of molecular events, gene maps, producing food with extra nutrients, nitrogen fixation, study of defective genes, tailor-made organisms, genetic disorders, and recombinant products. But recombinant DNA technology finds its maximum application in the field of agriculture and that of medicines. Now students, tell me, in your junior classes you have read about the various ways where how the food production can be enhanced. Just recall those, how the food production can be enhanced. It can be enhanced by three basic steps or three basic points where changes can make a drastic change in the production or the yield of the crop. The first is selection of high variety of seeds or high yielding varieties. What do I mean by high yielding varieties? Which give maximum production from the seeds. The second is changes in the cultivation technology, like use of chemical fertilizers, pesticides, etc. to enhance the yield. And third, post-harvesting management of the crops. So, in the process of producing the high yielding varieties of seeds, the conventional method of hybridization not only produces seeds with desired qualities, desired characteristics, but also produces seeds with undesired characteristics. Second, excessive use of fertilizers and pesticides have resulted in the process of biomagnification and accumulation of these pesticides and insecticides and fertilizers in human body, and that is why DDT is now banned, because it gets accumulated in the crops, which again gets accumulated and enhanced and magnified in humans. Now, nowadays organic farming is very much a popular concept, where only the organic or biofertilizers and biopesticides are used. These biofertilizers and biopesticides used externally are not that potent. That is why for the seeds that are used for organic farming needs to be of superior quality. And at the same time, we need different, you can say, agrochemical procedures to increase the crop yield. Now students, genetically modified organisms or genetically modified plants, to be called more precisely, are the best source of the high yielding varieties. These Genetically modified plants are so engineered that they have a variety of advantages for giving you a high yield of crop. Like, these are genetically modified plants or crops. These are resistant to abiotic stresses like your drought, temperature, etc. They also are resistant to post-harvest losses. These plants are so modified, they have maximum mineral utilization. They have enhanced nitrogen fixation. These have enhanced viral resistance. The mineral nutrition and the nutrients are also enhanced in these crops. Like the most prominent example, if I have to give, you all know when India became independent, the government was facing a problem to provide food to each and every citizen of our country, because the British had exploited our country to such extent that the poorer sections of the society were not able to feed themselves properly, and they were suffering from massive deficiency diseases. At that time, government had undertaken the projects like Green Revolution, White Revolution, Silver Revolution to increase the production. And being India an agriculture dependent country where 62% of the population is dependent on agriculture, still we were suffering from shortage of food. So it was the primary duty of the government to supply farmers with the seeds that were not only high yielding but also were rich in nutrition or nutrients. The golden rice, which was a genetically modified paddy crop or rice variety that was fortified with vitamin B12, and vitamin B12 deficiency was massive in the Indian population. So it was due to this rice plant, which was enhanced with the genes for vitamin B12 production, vitamin B2 production, that the India somehow came out of the deficiency of vitamin B12. So these genetically modified crops nowadays are more essential, because nowadays the cultivation land is decreasing day by day due to industrialization, spreading of the cities. So there's very small land available to feed such a vast country. So we definitely need the varieties of plants which are going to give a huge production, and that is not possible without genetically modified plants. At the same time, these genetically modified plants are given properties like insect resistance and pest resistance from inside, so that the cost of productivity decreases. There is no need of external pesticides, insecticides. This will not only enhance the quality of the crop but also save the farmers money. And the most prominent example of insect resistance introduction in plant is Bt cotton. This term Bt stands for Bacillus thuringiensis. This Bacillus thuringiensis produces a toxin which is toxic to most of the insects. What does this toxin do? This toxin, when it's secreted inside the bacteria, is secreted in inactive form as protoxin. But the moment it goes inside the insects, like the bollworm which infects the cotton, it produces pores in the or channels in the gut of the bollworm. As a result, the insect cannot take food in and dies within a short span of time. And this protoxin is activated by the highly alkaline pH of their gut. So, the cotton plant, which is oftenly infected by the bollworms, they are genetically modified to produce this Bt toxin. So, the cotton plants, each and every cell of the cotton plant is having this Bt toxin, and whenever the bollworms, they infect the bud of the cotton, thereby decreases the quality of the cotton produced. Whenever they infect the bud, they ingest this Bt toxin and are gradually killed. So, the genetically modified cotton plants are made with this particular genes of Bacillus thuringiensis toxin, and it gives resistance to the cotton plant against variety of the insects. Later on, it was not only introduced in the cotton plant but also in the potato, tomato, brinjal, etc., so that the insects cannot bore into these plants and the products are healthy and huge in amount. This Bacillus thuringiensis toxin does not harm us or the humans because neither our gut is that much alkaline to convert the protoxin into the toxin, and secondly, the acidic medium of our gastric juice destroys this protein or protoxin. Hence, we are safe from the toxin, but the insects that ingest the Bt toxin gets killed. So, it is only possible due to insertion of this toxin genes into the seeds of cotton, seeds of brinjal and other vegetables, so that the plants from internally they are resistant to these insects.